Welcome to Chapter 1 of Unit 1, the main themes of microbiology. In this chapter, we're going to learn about what microbiology is, who studies that and why, what are microbes, and more. So the definition you'll definitely need to know for your first test is what is microbiology? And the definition of microbiology is it is a specialized area of biology that deals with organisms that are just too small to be seen with the naked eye. That means unaided eye. So in order to see these, we are going to need a microscope and usually some chemical agent to visualize them. Microorganisms are often called microbes, and they include germs and viruses and agents or bugs. And contrary to what most people think, only a small amount of them actually cause harm to other living things. So there's cellular and non-cellular microorganisms. If you've learned anything in biology, all living things are made of cells. So cellular organisms are the living microorganisms. Bacteria, archaea, fungi, protozoas, and helminths. Now, helminths are technically not microorganisms, but they do cause human infection. They are very small, and these are worms. Yes, I said worms. Non-cellular microorganisms are non-living things. So technically, we could argue, are they organisms or not? But these are going to include viruses and prions and more on what those are. Now, microbes or microorganisms are easy to study, but they're also difficult to study. Why are microbes easy to study? Well, we can grow them very quickly in the lab, pretty easily. They reproduce at a rapid rate, which allows us to do that. But they're difficult to study. As I said, we can't see them, so we're going to need microscopes to see these microorganisms and use chemical tests. Microbiologists study a number of things about microbes, their cell structure and function, how they grow, what their physiology is, what their genetics are, the taxonomy, the naming of the microbe, and the evolutionary history throughout time. We also look at how these microbes interact with both the living and the non-living environment. There's a number of branches of microbiology. Your test will not be heavy on these, so I'm going to rush through them for you. But if you went into microbiology as a career, you could do a number of different things. You could go into medical microbiology, which looks at human disease. You could do epidemiology, which monitors disease. And you could work for the CDC, for example, or the World Health Organization. You could study immunology and look at vaccination or blood testing. A very popular field is to study immunology with allergies. Industrial microbiology is safeguarding our food and water in our industry. It includes biotechnology, where we use microbes to actually make products, everything from amino acids, beer, wine, cheese, bread, drugs, vitamins, lots of industry, including microbes. Agricultural microbiology, looking at the relationship between microbes and farm animals or crops. Environmental biology, how the microbes interact with the earth, and we could study the soil microbiology or aquatic microbiology, geomicrobiology, so that's in the earth, or even astrobiology. Microbes have shaped the development of the Earth's habitats and the evolution of other life forms for billions of years that microbes have been here. Soon after the Earth was formed, that first ancient cells started to form, and from those we see bacteria and archaea development. Later, much later, eukaryotes appear. They think about 1 billion years later. So cells with a nucleus are classified as eukaryotes. Eu means true and kary means nucleus. So eukary means a true nucleus. So any cell that has a nucleus is considered a eukaryote, and you do want to know that for your test. Cells that lack a nucleus are called akaryotes, a meaning no or without. And so remember, kary, Kary, is nucleus. So bacteria and archaea are living organisms, so they are cellular, but they don't have the nucleus inside. So here's a kind of a timeline that scientists have estimated the arrival of these different cells and organisms. So between 4 and 5 billion years ago, we think the Earth was created, and some pre-cellular structure was here not long after that, which gave rise to 3 different types of organisms: bacteria, archaea. Now remember, both of those are eukaryotes, or you probably heard prokaryotes, because if you'll notice, they were on the scene way before eukaryotes. So eukaryotes or prokaryotes are your archaea and bacteria, and that just means they don't have a nucleus. And then later, we see eukaryotes come into play. And eukaryotes are cellular organisms that do have a nucleus. And then insects, reptiles, mammals, and finally humans evolve. And all of these are types of eukaryotes. Insects, reptiles, humans, which are a type of mammal, have a nucleus. Bacteria are ubiquitous. And what that just means is they are found nearly everywhere. We see bacteria found in the Earth's crust, in polar ice caps, in volcanoes, in oceans, inside you, inside plants, and even other animals. Oh, excuse me. So what is the definition of evolution? We've mentioned that word a few times. This book's definition is the accumulation of changes that occur in organisms or in organisms as they adapt to their environment. The theory of evolution has been documented all over the world, and we see this phenomena and we have tested it in science. It is well studied and well established, and it is a natural phenomena. We don't focus on evolution in this course. There's evolutionary biology courses you can take, which discusses how that happens. So microbes are deeply involved in the flow of energy and food and how it moves through the Earth's ecosystems. In fact, without microbes, we would not have the cycling of matter. So they believe bacteria did invent the process of photosynthesis. Anoxygenic photosynthesis. So an or a in front of anything means without, and oxygen, that is the stuff you're breathing. Anoxygenic photosynthesis and the later evolution of oxygenic photosynthesis, bacteria used. So anoxygenic doesn't produce oxygen while oxygenic does. So eventually the production of oxygen by these microbes allowed other cells to develop and species to diversify. Photosynthetic microorganisms do contribute to, in a large way, how much oxygen is in the atmosphere. And what I think about ecologists and environmentalists who want to, you hear about saving the trees, right? All this information, save the trees, save the rainforest. And as a child, I thought they were worried about how much oxygen was in the atmosphere. But really, most of our oxygen in our atmosphere is accomplished by bacteria and algae. And so really we need to pay attention to the water sources also for photosynthesis to protect amount of oxygen in the atmosphere. Bacteria help with decomposition. They break down dead waste and matter. This is a pretty gross picture of a herd of some mammal decomposing, and bacteria and fungi both do decomposition. Microbes are so important because they're the main forces that drive structure and content of soil and water in the atmosphere. And of course, those are going to be important for other organisms. Microbes produce gas. Microbes, as we mentioned, live in the Earth's crust, and so they're in the soil. and they have relationships with other plants or other organisms like plants and animals. Humans have been using microorganisms for many years, manipulating them to improve our life. For example, yeast, a fungus, helps produce wine, beer, and bread, and cheese is another fungus that's used for that. So fungi have been being used for a long time. Moldy bread was used in Egypt to treat wounds, and so this is a major industry. And microbes helped, you know, they were at the center of it. Biotechnology is the manipulation of microorganisms to make products for industry, and recombinant DNA technology, which we'll study later, manipulates the genetics of microbes or plants and even animals to create new products, and we call those GMOs. Bioremediation uses microbes to restore stability in our environment and clean up toxic pollutants. For example, there's bacteria that eat oil. They break down oil. So during oil spills, one of the common things microbiologists and ecologists did was perform bioremediation by putting these oil-eating bacteria out at the oil spills to help break down the oil and get it out of the ocean. Let's talk about pathogens. You do want to know this for your test. The prefix path means disease, and gen, we see the word gen as in generator or the book of Genesis. It means to create or make. So pathogen is to create disease. So a pathogen is any agent, such as a virus, bacterium, fungus, protozoan, or even a worm, can cause disease. And there's 2,000 different microbes that can cause disease. Excuse me. So, if it's not caused by microbes, it is often non-infectious or non-communicable. But if it is caused by microbes, it may be passed on, communicable, and or is infectious. So the U.S. top causes of death of all disease, we see heart disease is number one, and cancer is number two in the United States. And then COVID-19 was also a large disease causing, a death causing disease. We have accidents, chronic lower respiratory disease, strokes pretty high along with Alzheimer's, diabetes, kidneys, and influenza. Heart disease is so high in the U.S. due to our diet mainly. But what is caused by microorganisms? COVID, flu, and pneumonia. So of all diseases, here's the world's top causes. So we see heart disease is a world problem, stroke, chronic lung disease, influenza, pneumonia, COVID-19. So that's deaths in 1 year, in the millions. Okay, and then neonatal conditions, respiratory cancers, Alzheimer's, diarrheal disease is very high in the world, not so much in the U.S., diabetes and kidneys disease. So which of these diseases are caused by microorganisms? Of course, we already mentioned COVID-19, flu, and pneumonia, but these diarrheal diseases, due to lack of clean water, also cause deaths. So here's a poster or a visual on communicable versus non-communicable diseases, infectious or non-infectious. What a great graphic. 30% of worldwide death are caused by infectious disease. So this is the change in death and disability rates from 1990 to 2015. So measles went down by 94%, diarrheal diseases 67%, but diabetes has gone up, probably because of our diet. So of course that is non-infectious. At the bottom, we see graphs of the United States versus the world and the current number of deaths. And we see that for the US, our non-infectious diseases cause way more deaths than infectious per 100,000 people compared to the world where there is more infectious disease. So, one of those diseases that interests me quite a bit is malaria. Malaria is a big killer in the world. It kills about 450,000 people every year. And this disease is transmitted by mosquitoes. I happen to take an immunology course with Harvard on malaria. Super interesting. The prevention of infection that right now they're doing to prevent malaria being spread, the most common one is bed nets. So literally a physical barrier. It's pretty inexpensive, it's easy to ship, and we can send those to the people in malaria infectious countries. By the way, there's other ways to help prevent malaria. There's some drugs, and there's also biotechnology that's being created right now, but that is way too expensive for many of the people in these countries. There are emerging diseases as well as older diseases that we see coming back: SARS, COVID-2, Ebola, AIDS, hepatitis C, encephalitis, polio, leprosy, and parasitic worm disease have been largely eradicated due to medical technology and education. There are some certain diseases that were once considered non-infectious but now are found to be caused by microbes. So this is really interesting. Gastric ulcers, they thought was due to stress. That might have some underlying conditions, but really what causes a gastric ulcer is a bacteria called Helicobacter pylori. Now, they have also discovered there's a link between certain cancers and bacteria and viruses. Coxsackie virus has been associated with diabetes and schizophrenia, MS, OCD, coronary artery disease, obesity are also linked to chronic microbial infections. So today we're discovering that there's this side of microbes that is slow and quiet and has gone undetected, or we've been unaware of as far as destructive diseases. For example, chlamydia infection does cause female infertility, and some liver cancers, specifically hepatitis viruses, can cause that, as well as the human papillomavirus, which can cause cervical cancer. An increasing number of patients with weak immune systems are subject to infections by common microbes that are not pathogenic to healthy people. So what we're saying is we see a lot of people with weak immune systems, either due to age or a primary infection, that can then get a secondary infection that a healthy human would not get sick from because their immune system is working and fighting those infections. Drug-resistant microbes have become a problem due to the increase of infectious disease and the abuse of antibiotics. So let's talk about general characteristics of these microbes. There are three basic cell lines that appeared, as we saw earlier: archaea, eukarya, and bacteria. Now, the majority of the microorganisms are single-celled, so all bacteria and all archaea, and only some eukaryotes are single-celled. All the worms and some fungi are multicellular. So we're just talking about microbes or microorganisms, things that we see under the microscope, okay? All animals, plants, most fungi, most not most, but some protists are multicellular, but all bacteria and archaea are single-celled, and there's some fungi that are and protists that are eukaryotes. Sorry, that are single-celled. Okay, so the bacteria and the archaeal cells, they are teeny tiny, really hard to see even under a microscope. They're 10 times smaller than the eukaryotic cells. And these prokaryotes or eukaryotes, remember, they don't have a nucleus. They don't have any organelles. And organelles are small double membrane bound structures. So we're talking about nucleus, the ER, rough ER, smooth ER, mitochondria, the Golgi, all of those things, not in bacteria and archaea. Eukaryotic cells do have the organelle. So remember, U means true and kary means what? Nucleus. Did you remember it? So eukaryotic cells do have a nucleus, therefore they have the other organelles as well. So like I said, the rough ER, smooth ER, Golgi, vacuoles, vesicles, mitochondria, and chloroplasts for those photosynthetic organisms. Acellular microorganisms, remember acellular, not made of cells, includes viruses and prions. So what is a virus? If it's not made of a cell, what is it made out of? It is a small amount of genetic material. So it's DNA or RNA, and it's just packaged in a protein cover envelope, they call it. So prions are small proteins that are folded in really intricate, complex ways, which behave like microorganisms and are transmitted from one human to another. So they can kind of turn on a disease. So here are the 6 types of microorganisms. Remember, helminths are worms, so this is the head of one. This is its scientific name. Sorry. So your scientific name's Homo sapien. This worm's scientific name is Tinea solium. And this is the scolex or the head of the worm. And that's kind of scary, right? And so this can cause disease in humans. Here's a fungus and a protozoan. Vorticella, a really pretty protozoan to see under the microscope. Bacterium. So bacterium is one. Bacteria is more than one. And so that's why they're pointing to multiple bacteria. But one of these would be considered a bacterium, and its name is E. coli, Escherichia coli. The virus, herpes simplex. So here is one particle of the virus. And then prions, which are mainly just proteins with these crazy shapes. We don't have any pictures of archaea here, but the archaea are also microorganisms. So we knew that foods would become spoiled and became inedible or would cause disease or illness. The Black Plague and smallpox we knew were caused by some kind of transmissible matter. Something was causing these. We really didn't know what that was a long time ago. And people believed in spontaneous generation. And spontaneous generation is this idea that living things can come from non-living things. They used to think that one of the examples of spontaneous generation is if you wanted to create little worms or flies, you made flies from meat. So you'd set meat out, and eventually little worms and flies would come from the meat. What they didn't know is that the flies that were landing on the meat were laying microscopic eggs, and those were developing into maggots and then casing into flies. But Louis Pasteur, who's super famous, disproved this idea that living things can come from non-living things. And so he left some broth out. Now, normally if you leave some soup out, right, like it's going to grow a film on it. It's going to become disgusting, and you can't eat that. It will get you to be very sick. So Pasteur did this experiment where he boiled the broth in this flask, which killed any microbes that were in it. destroyed them. And it had this gooseneck flask here. And so they thought there was something in the air that could cause the broth to go bad, but he's showing that air could still get inside, but any components in the air, like bacteria, would get caught in the neck here. And so what he found is there's some broth that, as long as bacteria, fungi, microbes cannot get to it, it will stay sterile. And I hear in a museum somewhere in Europe that there is still broth sitting from Pasteur's experiment. Okay. So as long as you keep it in, not open to microbes, it will stay sterile. And so here he showed like that if you broke the neck off, that growth will occur. But as long as you trap the microbes in the base here, like I mentioned, air can flow here. It's not air that's causing the broth to go bad. It's actually microbes in the air. So Robert Hooke, another really famous scientist, had a skill for grinding lenses. And so he made these convex lenses and ended up making a machine with double lenses that we now call a microscope. And Robert Hooke was looking at everything under the microscope. He was the first one to observe things that no one else could see. This, can you imagine, like all of a sudden there's a whole world open to see that we never could see before? Like he was looking at his hair, he was looking at fingernails, he was looking at skin cells. And he said, oh my gosh, there's these little bitty things that are growing and living. And he actually helped develop the cell theory from that. Now, Antoine van Leeuwenhoek used these microscopes, and he was examining threads and fabrics and things. And he called these little organisms that he saw animalcules, because he was looking at rainwater and he saw little things moving around behaving like animals, and he called them animalcules. He also looked at scrapings from his teeth, which is pretty gross, if you ask me. Now, we've gotten a lot better at microscopes since Hooke and Leeuwenhoek. We've refined our lenses. We have finer focusing devices with built-in light sources. They used to have to use mirrors to angle the light. And so we can see a lot now, even as technology has advanced, than they could back when Hooke and Leeuwenhoek invented the microscope. So in the 1980s, they invented the PCR technique. PCR allows us to take a sample of DNA and replicate that sample 1,000,000 times within a day. So this was huge for really genetic testing and criminology. We were able to collect DNA from crime scenes, make millions of copies of that DNA to investigate it. And this was key because think about, if you have a crime scene, there's only a little bit of DNA. If you do experiments on that and ruin that DNA, you just ruined all the evidence. But PCR allows us to make copies of that DNA so that we can do bunches of experiments with it. In the 2000s, we learned the importance of small RNAs, like RNAi. And then we were able to do the Human Genome Project and look at not just the human genome, but the microbiome of humans. In 2013, we came out with CRISPR, which is genetic editing using Cas9. So we can essentially, just like if you were to type out a paper and you see one word you've made a mistake in, you can delete that word and replace it with another. We can do that now with genes and DNA. And so science is forever evolving and getting new information, and technology is going way faster than we can keep up, which is problematic for me because constantly learning new things as a biologist. You know, I hate to say history doesn't change. I guess views of history changes, but like math stays math. Biology is a tough subject to study because it is ever changing. We're disproving old ideas all the time. And we're using the scientific method to do that, which is this general approach taken by scientists to explain what happens in nature. Now, a hypothesis is not a guess, it's actually an explanation. So many teachers misteach the word hypothesis. It is an explanation for what has been observed or measured, and it has to be testable. You can either support it or you can discredit it. It is really, a hypothesis is never proven ever. It's only supported or unsupported. And that has to be done through observation and experimentation. So here's your book's general idea of a scientific method. You have some question that you want to answer, and you do some background research. You're studying what other people have thought. You look at other experiments that have done, and you might construct a hypothesis, an explanation, and then you test it. And usually you have a hypothesis and then you actually have a null hypothesis. And you test those hypotheses with experimentation. You recollect data, your own data. You either reject your hypothesis and accept your null or vice versa. And you might, if you reject your hypothesis, you might construct a new hypothesis and run new experiments to test it. But whatever you're doing, you want to communicate those results so that you can share with the scientific community what you've discovered, and they can use that information for their own questions. So it's not a step one, step two, step three. It's kind of a web of a process. Now, deductive reasoning is using general principles to explain specific observations, and then the kind of the opposite is inductive reasoning, where you discover these general principles by carefully examining specific cases. So you're getting little, small, minute, specific ideas from larger ideas, or you're using smaller ideas to create the larger ideas. In inductive reasoning, you are making observations through experimentation, which often leads to a discovery of a principle or general idea. So, for example, here's a great way to compare deductive and inductive reasoning. Deductive reasoning, it says knowing that opening attachments from unknown senders can introduce viruses or other bad things to your computer, so you choose the specific action of not opening the attachment. You've got this general idea, so you don't do the specific thing. Inductive is you've performed the specific action. You clicked on some attachment 3 different times, and each time your computer crashes, and that leads you to the idea that you should probably not open attachments that are unknown because they could damage your computer. Again, the scientific method is this lengthy process of experimentation and analysis of your data, and then further testing. It eventually leads to conclusions that you either support or refute your original hypothesis. As I mentioned, if experiments don't uphold your hypothesis, the hypothesis or some part of it has to be rejected, and it's either discarded totally or modified to fit your hypothesis. So if your teachers ever did claim evidence reasoning, this fits in with the scientific method very well. If your experimentation does support your hypothesis, it like I said, it's not proven, it's not accepted as fact. It has to be tested and retested, usually by other people. The experiment needs to be replicated and the results must be replicated. And then that information is published and repeated, as I said, by other investigators before we really accept it as a fact. So we've talked about a hypothesis is not this guess, but it is an explanation of what's going to happen. A theory is a collection of statements and propositions, all these concepts that describe what's happening in nature. It is a huge body of ideas and explains this phenomena, and it is been tested and retested by many, many people. It always occurs. It stands the test of time, and it has never been disproved by any serious scientific endeavor. And so when we talk about theories, there is no evidence. Remember, science uses physical, testable evidence to decline or disprove a theory. And if it ever does happen, they'll have to replicate those results again and reevaluate and figure out what's going on. So science and its hypotheses and theories must progress with technology. As we improve technology and the machines that we're using, we have to re-examine these old theories and old hypotheses in order to propose new ones. So usually scientists are curious and open-minded. They are skeptics. They are usually creative, and they want to cooperate with one another and work together. And they are willing to revise their views based on new discoveries made. Sorry about that. So early experiments showed that microbes are everywhere. Air, dust is full of them, entire surface of the earth, all the bodies of water, all the living things inhabited by them. John Tyndall found that microbes are in dust and air, and many of them have a high heat resistance. Later, the discovery and detailed description of specifically heat-resistant bacteria, endospores, clarified that heat-resistant microbes exist. Now, what is sterile? That just means it's completely free of all forms, including spores or virus particles. Robert Koch linked a specific microorganism with a specific disease. And Dr. Oliver Wendell Holmes observed that mothers who gave birth at home experienced fewer infections than mothers who gave birth in the hospital. And the next person showed that women became infected in the maternity ward after examinations by physicians who had been working in the autopsy rooms without washing their hands. So all of these people observing these phenomena helped develop aseptic technique. Now, Joseph Lister was the first to utilize this idea of hand washing and misting operating rooms with antiseptic chemicals. His techniques became the foundation for modern microbial control that is still used today. Washing your hands is so important, as you know. Remember Pasteur? He invented the process of pasteurization, which we pasteurize our milk. And this showed that human diseases could arise from infection. When he pasteurized things, we got rid of some disease. Now, Robert Koch established a series of these proofs that verified the germ theory of disease. So we now learn that diseases are coming from microbes. We should wash our hands. We should use aseptic technique. Now, nomenclature, as I mentioned, is the assignment of a scientific name, naming organisms, using categories and individual organism names. And we can classify them. And this is an attempt that is constantly changing right now, to orderly arrange organisms into a hierarchy of taxa or categories. And this classification is difficult because, like I said, as technology improves, we learn more about the organism. We find out, these organisms are actually more different than we thought, so they don't need to go in the same category. And the process of discovering and recording traits of an organism so they can be recognized or named and placed in a category is identification. We use a binomial nomenclature system. Bi means two, and nomial means name. So it's a two-name naming system. And it uses the combination of your genus name and your species name. The genus name is always capitalized, and the species name always begins with a lowercase letter. So it is not capitalized. And both names, when typed, should be italicized, but when you have to write them, you underline them. So, for example, Homo sapien, your scientific name, which word will be capitalized? Homo or sapien? If you said homo, you're correct. And which word is your species name? Homo or sapien? Sapien is your species name. Homo is your genus name. And we do abbreviate these. So Homo sapien can be abbreviated H period sapien. And so we see this with Staphylococcus aureus. It can be abbreviated to S. aureus, and that is a type of bacteria. So when we talk about taxonomy, the naming of organisms, there's different levels. And so there's a way to remember this. Dumb King Philip came over from Germany sober. There's not a king of Germany, but he thought he was the king of Germany. And he was always drunk. I don't know. You could say drunk King Philip came over from Germany sober. Really weird for drunk King Philip to be sober. So that's how I remember this. You can come up with your own way. But this is going from the most general, the largest group, to the smallest. Have you ever seen those like Russian nesting dolls? Or have you ever gotten a huge box for Christmas only to find that inside that huge box there's a lot of little boxes, and in those little boxes there's even smaller boxes, in those smaller boxes even, that's kind of this thing. It's a hierarchy. So domain is the largest, the most inclusive category. And inside a domain are different kingdoms. And inside one of those kingdoms are different phylums or divisions. And inside one phylum is a bunch of classes. And inside one class is a bunch of orders. And inside an order is a bunch of families. And in a family, there's genuses. And in a genus, there are different species. And so this is a great example of Homo, our genus name, and sapien, our species name. So notice Homo is capitalized down here at the bottom, and sapien is lowercase, and they are italicized. So there are other homos, if you're wondering. They're just not alive. There's Homo erectus, Homo habilis, and then Homo sapien. And there's other homos. Those are the ones I know off the top of my head. But let's look at our most inclusive level, way up here, the domain. The domain eukarya includes all eukaryotic organisms. Remember what that means. They all have a nucleus. They all have organelles inside their inside their cells. So, who belongs in this domain? Whatever this is, it looks like maybe a plant, all these animals, these protozoa, some algae, all of these are in the same domain. It includes a lot of organisms. But in kingdom, dumb King Philip came over from Germany sober. In the kingdom, we have the animal kingdom. Within the animal kingdom, we have the chordatas. Now, a chordata is any organism that has a notochord and this dorsal nerve cord, and usually pharyngeal slits as an embryo. And so this is anatomical structures we see in all of these organisms that are in this phylum. Now within that chordata, there's a bunch of different organisms, but specifically there are different classes, but one class is called Mammalia. And we belong to the class Mammalia, as well as dogs and gorillas, and maybe that's a lemur. I don't know my organisms like I should. But all mammals have hair and mammary glands. Now within class Mammalia, there's a bunch of orders. And Primates is one of those orders. And so we belong to the order Primates along with the lemur and the big gorilla. All primates have digital dexterity, meaning they can move their thumb around. They have large cerebral cortex. They have a slower reproductive rate, a longer lifespan, and forward-facing eyes. I'm surprised that's not written. Within the order Primates, there's Hominidae, and we belong to that family along with the great apes. We have a larger brain, we don't have a tail, and we have long upper limbs. But in that family of Hominidae, currently there's only one genus alive, and that's the Homos, and their species is Homo sapien. We can do the same thing for the kingdom Protozoa and come down to this Paramecium caudatum, a really popular microorganism, a protist. So it's kingdom Protozoan, phylum Ciliatea, class, let me try this, Hymenostomia, Hymenostomata, Paramecidia, Paramecium, and caudatum. Woo. Taxonomy uses Latin and Greek language, and it's really difficult for me. So as I've been just using this taxonomy, this is the process or science of classifying biological species, naming them, putting them into these taxa, these groups. And Carl von Linné in the 1700s came up with these basic rules to follow and established these categories. And we've used them ever since to organize all of not just things that are alive now, but things that are extinct. Now, phylogeny is the taxonomic scheme that represents natural relatedness between groups, and it is based on evolution. And so you can see that our last common ancestor, whatever organism this was, gave rise to bacteria, archaea, and eukarya, they believe, based on genetic and morphological studies. So we have this universal tree of life, and it includes the 5 kingdoms. Your 5 kingdoms are plants, animals, protista. It used to be Monera and fungi, and I'm surprised they're using word Monera. We'll go with it, but know that that is changing as we speak. Molecular genetics allows, remember, looking at the molecules and the genetics that create the organism, so proteins and DNA and RNA, allowed us to study the structure of different types of RNA, and ribonucleic acid in a small subunit of our ribosomes was discovered that really helped us learn that there are 3 main domains: bacteria, archaea, and eukarya.